All right, so today we're taking a look at 8.2, which is the cell cycle. So cells, it's kind of like their lifespan. They go through this series of growth and they're preparing to divide, and then finally they do divide. And that whole cycle, they repeat it then. So once they divide, they go through it again. That whole cycle is called the cell cycle. Again, it's like a life cycle. You and I have a life cycle. We're born, we grow larger, reproduce. Cells do the same thing. Cells are going through this cycle, growing, preparing to divide, and finally dividing. So last week on their notes, we got to get kind of artistic and we drew some boxes. Today, getting artistic again, and we're drawing as best as we can a big old circle. I'm not the best artist, you guys can tell, so do your best, draw a big old circle. But if it's a little, oblong on this side, little flat on this side, hey, that happens. Mine is too. And we're going to go ahead and divide this circle into quarters. Now I'm really going to get artistic, and I'm going to draw a second circle outside the first circle. But not all the way around. In fact, when I get up top here, I'm going to kind of do a little trick. Hopefully you guys are using pencil, not pen. Erase a little bit and turn it into an arrow. So the arrow covers 3/4 of our circle, 3/4 of our cell cycle. This arrow represents, so it covers all 3 of these phases. This arrow represents what we call interphase. And that's where we're going to start. Actually, I'm going to start just a line or two below. I'm going to leave myself just a little bit of room up here. So interphase. So interphase is so named because it is the phases, is the phase of the cell cycle between the phases of mitosis. When you look at cells through the microscope, it's actually really easy to tell when they're in a phase of mitosis. There are things happening, there are defined pictures or defined images. And so for a long time, the scientists basically said, hey, if it's not in mitosis where something's happening, then it's in between mitosis. Inter means between. It's Greek for between. So we are between the phases of mitosis. And although it might look like there's not a whole lot going on, there really is. It really is. So the three phases of interphase, we're starting this one here, beginning of a cell's new life. This is called G1 or growth 1 phase. Now, some people call it gap 1. I like the name growth better because that describes what's happening. The term gap 1 is from back when we didn't think there was a whole lot going on. It's just a gap phase, but that's not really accurate. There is quite a bit going on in this first stage. So during G1, during first growth phase, the cell grows and replenishes organelles. You have to think, hey, that cell just divided. It just came off of division. It's a brand new cell. And so it needs to grow. It needs to get a bit larger. It needs to become a grown-up cell, if you will. So it replenishes its organelles that it lost when it divided. And that's what's going on here in G1. Questions? Taking a quick pause here. Good time to ask for questions. Taking a quick pause because I got to turn the page to continue the notes. So anything you guys want to ask while I'm paused and letting people catch up? All right. Well, let's flip the page then. Well, actually, we're doing this quite a bit today, back and forth. Feels like you got more than one page here, but perhaps not. All right, G1 phase is followed by synthesis phase, which we usually just call S phase. And as the name kind of implies, synthesis means to make. So what are we making? Well, during S phase, that's when the cell duplicates, or again, synthesizes. There's the term. There's why we have the name of the phase. Synthesis is to make. We're making new DNA. We're copying our DNA so that when the cell divides again, both of the new cells can have an exact copy of the DNA. All right, let's flip back to our diagram. G1 is followed by S phase. And it is during S phase that we have DNA duplication. That's where the DNA is copied. Now, this is kind of interesting. It's kind of weird. Maybe a fault of science. I don't know. Important, not just for this chapter, but for the next several chapters. When we think of a chromosome, I think we usually think of kind of an X shape like that. I think a lot of people think of chromosomes as having an X shape, and that's good. That's accurate. That is a chromosome. That is definitely a chromosome. But here's the thing. That is a duplicated chromosome. Prior to S phase, chromosomes are just kind of a single strand. They're not an X. They're just a single strand. And it's during S phase that one strand copies itself and becomes two strands that kind of make that X shape. And again, here's the problem we have with science. This is called the chromosome, and this is called the chromosome. Got the same name, and yeah, they are the same thing. They're both chromosomes of DNA, but this one has twice as much DNA. You know, in English, we kind of took care of it. Like, you don't have just one sock. You fold over the top so that your socks are stuck together. You know what I'm talking about? You keep your pair of socks. Well, we have terms for that. We distinguish between sock and socks in English. In biology, for whatever reason, we don't. This is a chromosome. This is also a chromosome. So kind of confusing terminology. Again, we're going to work with it for the next several chapters, so bear with me. But know that it's here during S phase that strand of DNA copies itself. And then, again, like socks, they stay stuck together in the middle. And that's why it looks like an X. They'll get pulled apart later during mitosis. So we get to mitosis, we'll watch them get pulled apart. But for right now, we're going to keep them stuck together. Questions about the terminology there? Cool. All right, let's go back, continue the notes. After S phase, we have a second growth phase called growth 2, or usually we just symbolize it G2. And as the name implies, growth, during the second growth phase, the cell is going to continue to grow and it's going to prepare for division. It's getting ready to divide in two. So it has to make sure it's large enough. It has to make sure it has enough organelles for two new cells to have. So we've got a lot of growth and preparation going on during G2. This is also where we form asters. So asters are formed during G2. Have a question through the chat. Very good question. Someone asks, how long is the cell cycle? And it kind of depends on the type of cell. So bacteria, for instance, now they don't really go through the cell cycle like we're talking about, But really simple cells like bacteria, they can divide every 20 minutes. So that's pretty impressive. Most eukaryotic cells, and again, our cells are much more complex. We've got a lot more organelles, a lot more DNA. Again, it depends on the type of cell, but generally between 24 and 48 hours. So a cell can divide every day to 2 is what most cells, how long they go through the complete cycle of the cell cycle. All right, a note on asters. So asters is a Greek word. It means stars. So think of like the word asterisk. You know, like if you put an asterisk next to something, kind of looks like a star, right? Well, it's because in Greek it is star. So asters are these radiating microtubule growths. If you guys remember back to last semester, we talked about microtubules being a part of the cytoskeleton. It's almost like the bones of the cell. So these asters are these growths. So these microtubules are growing, they're radiating out from the middle. And they'll have a very important role to play when we get to mitosis, which we don't get to until Friday. But when we get there, you guys will see that they're really, really important. We're getting all kinds of wrong terms here. Sorry about that. And I'm still spelling it wrong. Bear with me. All right, in animal cells, the asters are grown from the centrosome. And the centrosomes are comprised of 2 centrioles. Centrioles usually look like little logs. I'm not very artistic, so you won't see it when I draw it, but that's kind of what centrioles look like. That in animal cells, it seems as though these centrioles are building the microtubules, and that would make sense to us. They're building these long proteins that are coming out from the centrosome. But here's the weird thing. In animal cells, again, it seems as though the centrioles are building the microtubules. And we wish it were that clear and cut and dry, and in animal cells it kind of is. The problem is plant cells don't have centrosomes or centrioles, yet they still make asters. So they're still building those microtubules that are radiating out, and again, have a very important role to play in mitosis. We'll see that in a couple days. So it's not, the formation of those asters is just not as well understood as we would like to think. If centrioles play such a big role, why is it that plants can build them without centrioles? Hey, that's a lot of terms. A lot of new terms for you. And I think pictures worth 1,000 words. So let's take a look at a quick little diagram here. There's an animal cell. Here's a plant cell. I'm going to give them both a nucleus. So they both have a nucleus. In the animal cells, we're going to go ahead and put a pair of centrioles into a pair of centrosomes. So what I mean by that, and again, these little circles here, they're supposed to be almost like log shapes. I'm not a very good diagrammist, so I'm not going to draw the full log, but that is what we call a centriole. The pair of them make up what we call the centrosome. So, better eraser. So the two centrioles together make up the centrosome, and then radiating it out from that area is the aster. Now we're actually going to have a pair of these. We're going to have two of them. That's our aster. Again, you can kind of see why it looks like a star, or why it's called a star. We've got these little microtubules that are starting to grow out from these centrioles. Oh, I spelled centrioles wrong. I'm sorry. Man, rough day already. Forgot the N. Sorry about that. Hopefully you guys have an eraser if you also misspelled centriole. Sorry about that. C-N. All right, so in plant cells, it seems to be, to us, that the centrioles are building or forming those asters, are pushing those microtubules out. Plants, though, they don't have centrioles. They don't have centromere, centrosome. But they do have asters. So why are their asters forming if they don't have centrioles to make them? We don't know. Not entirely well understood. Questions? All right, let's flip back. So this third phase of interphase is called G2. And again, that's where we have growth and preparation for division. We are getting ready to divide. That's going to be our next step right here. It's division. And also, it's during G2 that we have our aster formation. All right, while we're on this page, well, let's go ahead and build our last quadrant here. Go ahead and add that double line, make it an arrow again. Hopefully you guys have an eraser on there. So I've got this second part of the cell cycle, and this second part is called M phase. M phase is made up of two parts itself, so I'm actually going to divide this area into two, and then with this bottom part here, I'm actually going to take up a little bit. They overlap. This bottom part overlaps with the top part. There's not a clean break as to when this one ends and this one begins. So I've got that kind of overlap segment right there to show that they both kind of happen at the same time right in here. This top portion, I'm going to go ahead and divide this into fourths. Because there are 4 parts to this top part. Dirty eraser. All right, well, let's go talk about M phase. So flipping back to the previous page. M phase. So during M phase, the DNA and the nucleus divide, followed by the cell dividing, the cell itself dividing. So that's why we split that second part into 2. There are 2 very distinct parts to M phase. The first of those 2 parts is called mitosis. Mitosis, very specifically, is the division of the nucleus and the DNA. Really drives me nuts when people try to refer to mitosis as the entire cell cycle. And I hear it all the time. Mitosis is definitely not the cell cycle. We see it on that other page. It's actually just a small little portion of the whole cell cycle. In fact, mitosis is not even the division of the cell. Mitosis is merely the division of the nucleus. Now, that's a really important part of the division of the cell, but it's not the entire division of the cell. Mitosis is the division of the nucleus and the DNA. All right, the 4 phases of mitosis are prophase, metaphase, anaphase, and telophase. I always remember PMAT, P-M-A-T, and that helps remember the order of them: prophase, metaphase, anaphase, telophase, PMAT. So we flip back. This part right here is mitosis. Again, the entire cell cycle is definitely not mitosis. Mitosis is just one little part of the whole cell cycle. Mitosis has then 4 parts to it. Prophase, metaphase, anaphase, and telophase. And again, this last part kind of overlaps, especially with telophase, which is what we drew there. Got that overlap. P-M-A-T, PMAT. All right, one last little bit. Gotta make sure it's on there, don't I? Cytokinesis is the second part of M phase, and that's the division of the cytoplasm and cell. So that's the rest of the cell kind of pinching in half and going from 1 to 2. Again, mitosis is really only the division of the nucleus in DNA. Cytokinesis is where the rest of the cell splits in half. One more flip back. And there we go. Cytokinesis follows mitosis. It's the second part of M phase, and that's where the cell divides in 2. And the new daughter cells can re-enter G1. They can start growing again and go through the whole cell cycle all over again.